Okay, we're going to look a little bit more deeply at adaptive immunity in Chapter 16. We'll talk about primary and secondary antibody responses to infection and how this tracks to stages of disease. We'll talk about ways an antibody can participate in adaptive and innate immunity. Talk about how T cells work within the context of our humoral immunity and our cell-mediated immunity, and how the immune system avoids self-reactivity and how some pathogens take advantage of these mechanisms. We'll talk about the difference between humoral and cell-mediated immunity. We'll explain what an antigen, epitope, immunogen, and a hapten are, and we'll talk about clonal immunological response to an epitope and define antigen presentation and antigen presenting cells, or APCs. Okay, so innate and adaptive immunity, right? The basic difference with innate and adaptive immunity is that in innate immunity, we're not basing the attack against the pathogen on a particular specific feature of the pathogen. And because of the nature of the weapons used in innate immunity, sometimes we generate damage to healthy tissue, right? In adaptive immunity, we utilize the antigen present on the surface of the pathogen as a way to sculpt an attack that targets only that pathogen, and all the other white blood cells that could participate are left alone, right? And that makes the effectiveness of the system far more potent, okay? In adaptive immunity, we have slow development, we have cross-regulated defense, and we have memory, right? Once we tackle a particular strain of a pathogen with our adaptive immunity, we generate memory cells that stay with us our whole life, and then the next time we encounter that exact same strain of pathogen, we don't get sick because we have this standing army ready to jump the pathogen before it gets a chance to produce damage. Okay? In humoral immunity, we use antibodies as the mechanism of defense. These are just proteins that can attach to foreign antigens. An antigen can be anything from a protein, a glycoprotein, a lipoprotein, a carbohydrate. It could be any number of collections of molecules on the surface of the pathogen. Okay, and the antibodies get connected to their antigen, and when that connection gets made, that anything that antigen is attached to is going to be destroyed. Okay, so it acts primarily on antigen that's out in the open, all right, not inside an infected cell or on the surface of an infected cell. Plasma cells are what produce the antibody. They're derived from B cells that have encountered the antigen that they recognize and then have been activated by interaction with the helper T cell. Okay, T cells have the ability to recognize the antigen, stimulate B cells and cytotoxic Ts, and destroy infected hosts. Okay, there's different classes of T cells, just like there's different classes of B cells, right? Classes of T cells include cytotoxics, helpers, regulatories, and memories, right? The classes of B cells include your plasma cell, right? Your memory B cell, okay? And I think that's it, right? Just those two. So an antigen is just something on the surface of the bacterial cell or whatever pathogen we're talking about that has the ability to produce an antibody response. Okay, and in order to do that, it has to have certain characteristics and be of a certain size. Okay, not anything that is alien to our body will necessarily produce an antibody. Okay, for instance, if it's too small, it won't do it. Okay. An epitope is a binding site on the antigen, and so an antigen can have multiple epitopes. And a hapten is a foreign molecule that only becomes antigenic when it's attached to a larger molecule. Okay? In cell-mediated immunity, what happens is that helper T cells encounter on an antigen-presenting cell a foreign antigen. Okay? The T cell receptor interaction with that foreign antigen on that antigen presenting cell stimulates it, activates it, okay? It is then able to produce chemicals that can finish the activation of cytotoxic T cells that have encountered their antigen, and by direct interaction with B cells that recognize that antigen, it can fully activate those B cells so that they become plasma cells. That's why the helper T cell population is so critical in immunity, right? Once activated, the cytotoxic T runs out and finds infected cells and tissues and destroys them, okay? It's most effective against intracellular bacteria and viruses, which are going to be manufactured inside the infected cell. Humoral immunity requires the antigen-presenting cell, which would be a macrophage, a mast cell, a dendritic cell, or in some cases a B cell, an MHC complex, activated T cells to differentiate B cells into plasmas, and activated B cells, which generate an antibody that recognizes a particular epitope on an antigen. And during that process, the production of memory B cells that can spring into action in the event that we encounter that antigen again. Okay? So let's look at humoral and antibody-mediated immunity.
Humoral immunity is a type of adaptive immunity in which antibodies are produced to directly target microbial invaders. The antibodies are produced and released by plasma cells that developed from antigen-specific B cells. The antibodies attach to antigens on the microbes. An antigen is any molecule that will, when introduced to a person, elicit the synthesis of antibodies that specifically bind the antigen. To understand B cell activation and antibody production, a few backstories are necessary. First, B cells are born in the bone marrow. In these progenitor B cells, the genes that encode antibodies rearrange, allowing each mature B cell to express antibodies with a particular antigen-binding specificity. For a robust response to most antigens, the action of T cells and antigen-presenting cells is required. At the site of an infection, antigen-presenting cells, such as dendritic cells, engulf some of the microbes and digest them, putting the microbe's antigens on the cell surface, nestled in molecules called MHC2. In this way, the antigen-presenting cell advertises the antigens of the invading pathogen. The antigen-presenting cell travels to lymph nodes where B and T cells congregate. A T helper cell binds to the presented antigen with its T cell receptor, providing the first signal that activates the T cell. Note that the genes for T cell receptors undergo rearrangements similar to those for antibody genes in B cells, with each cell producing T cell receptors with particular binding specificity. T cells also have a co-receptor called CD4 that binds to the MHC2 protein itself. The engagement of other cell surface proteins provides the second activation signal, turning the T cell from an inactive Th0 cell into an activated Th cell, or helper T cell. In addition to producing antibodies, B cells are also antigen-presenting cells. They take in and digest some of the antigens that their antibodies bind to, and then display them on their surface with MHC2 proteins. When a T cell receptor of an activated T cell binds to the presented antigen, the B cell receives its first activation signal. The binding of two more proteins on the B and T cell surfaces triggers the second signal needed for B cell activation. Now activated, the B cell undergoes clonal expansion, resulting in a large number of B cells that can produce antibodies specific to the particular antigen. Some of these B cells differentiate into antibody-secreting plasma cells. Others become memory B cells that patiently wait to be called to future sites of infection, where they will generate a rapid antibody response. Antibodies bind to their antigenic matches, and by coding them, the antibodies flag them for destruction by other cells of the immune system.
Okay, we'll talk about antigenicity, okay, and we'll talk about specificity and its application to vaccines, okay. So antigenicity is just a measure of how well an antigen causes the production of antibody, right? Some examples of different antigenicities include this scale ranking nucleic acids versus carbs versus proteins, okay? But again, size considerations come into effect here as well. The threshold dose is the amount of antigen needed to produce an optimal response. A low dose doesn't activate enough B cells, and a high dose can lead to tolerance, okay? The idea of antibodies generated specific for a single antigen is a little bit like a lock and key, okay? Um, the idea here is that every antibody contains at least 2 binding sites, okay? And that means that the antibody, when it encounters the antigen, can essentially link multiple antigen molecules together and make them vulnerable to attack or cloak areas that cause pathogenicity, okay, like docking proteins in viruses or exotoxins, okay. It was Jenner that came up with this idea that we could expose the immune system to antigen in order to prepare it for an encounter with the real pathogen. All right. So what Jenner did was he took the exudate from cowpox pustules, and then he used that to inoculate people and produce cross immunity against smallpox. Okay. So the idea was that there are antigens on the cowpox that are similar enough to the smallpox antigens that the antibodies produced when the cowpox antigen was introduced. Now, this is not the live pathogen. This is either heat killed or it could be basically a breakdown product of the pathogen. So they're not giving you the disease; they're just giving you something that's disease associated, and then that causes the response of the immune system. We generate those B cells and cytotoxic Ts that are able to deal with that antigen in the event they see it again. And then when you encounter the real pathogen, you're able to destroy it before it really gets a chance to make you sick. Okay, and this is called artificial acquired active immunity. Okay, it's artificial because you're artificially exposing yourself to the antigen through an injection. It's active immunity because you are producing the white blood cells that are going to provide the protection, and that's going to happen throughout your life. Okay. Cross protection works between shared epitopes on different viruses, so that's one of the reasons why sometimes when people recover from infection A, they also end up having some immunity to infection B because the antigen in the 2 pathogens is similar enough that there can be binding of the antibody directed against A on antigen B. Okay. We'll distinguish immunoglobulin proteins from the family of immunoglobulins, diagram structures of antibodies, and talk about what they do, and talk about isotypes, allotypes, and idiotypes of antibodies. Okay. So an antibody is made of 2 long and 2 short chains, each of which have a variable and a constant region. Okay. The amino end of the heavy and the short chains are what are called the variable regions, and that's where the antigen binding takes place. The short chains and the long chains are held together by disulfide bonds, and they're kind of twisted around each other. And the class of antibody is determined primarily by the carboxyl end of the heavy chain, okay, which can designate it as going to exist as a free-floating antibody in tissue fluid, or maybe bound to the membrane of a B cell, or perhaps in tears or saliva or milk, okay, or perhaps present as a dimer or a pentamer, okay. But it does not affect, the constant region does not affect what antigen that antibody binds to. It's always going to bind to whatever the variable region says it's going to bind to. Okay, it's just that the constant region designates different locations for those antibodies to operate in. Okay, the different classes of antibody are shown here. Okay, IG, A, E, G, D, and M. Okay, the light chains can be either kappa and lambda. Okay, and then the heavy chains are going to have a constant and a variable region, okay, and a hinge region. The constant region of the antibody is the part of the antibody that reacts with complement or binds the surface of a cell, okay? So this is the other end of the molecule away from where the antigen binding takes place. The purpose of interacting with complement is to promote destruction of whatever the antibody is attached to through what's called the complement cascade, okay, which will produce, among other things, inflammation. But sometimes the constant region of the antibody is used to anchor it to a membrane surface, okay. For instance, it can serve as a B cell receptor in some cases, or it can serve as a way to activate a mast cell so that it degranulates and releases large amounts of histamine. Okay, so what different antibodies do what? Okay, well, IgG is your classic antibody. Okay, it looks kind of like what's shown here, and it's what's produced after the infection, the first infection, has sort of been around for a few days, okay? And it can activate complement, produce immunoprecipitation, and is found primarily in tissue fluid and in blood, okay? IgA is found in milk and in colostrum, is a dimer, okay? Does not activate complement, okay, but can bind foreign antigen. And so its purpose is to provide for the baby what's called natural passive immunity during the time that it nurses, okay. IgE, all right, can bind to the surface of mast cells and is active in allergic responses, which are basically releases of histamine that are designed to promote inflammation, which is designed to destroy the area where the pathogen could be coming into the body to cause trouble. Okay, but this sometimes results in healthy tissue damage in the event that this response is too vigorous. Okay, IgEs are they look basically like the molecule pictured here. Okay, IgMs are pentamers. They are the first antibodies that are produced during an infection process, and they intercept antigen. They can activate complement, okay, but they're less effective at doing so than IgGs are, okay. And then finally, IgD is B cell receptor. So it sits on the surface of B cell, binds antigen, gets internalized, right? And then B cell can take that and show it off on its surface with an MHC so that it can be fully activated by direct interaction with a helper T cell. Okay, so that's the skinny on most of those. All right, so the only ones that are present in more than one copy are the IgA as a dimer and the IgM as a pentamer. Okay, the rest are monomeric units, like is shown here. Okay. The variable amino acid sequences provide the antigen binding. Okay. The antigen binding site can bind peptidoglycan only versus another antigen, but it can bind other things as well, depending on its composition. Okay. The variable regions for the light chain are called V sub L, and the variable for the heavy are V sub H. The binding is reversible. Non-covalent bonding primarily and hydrophobic bonding guide the interaction, while the constant region can bind receptor on host cells or can bind complement, okay, in the blood. So you can see what some of these look like, right? That right there in the middle of the top, that's IgA, that's IgM. That could be IgG, okay? And then you can see some IgE here, right? Activating a mast cell, okay? Getting internalized, all right? So, you know, different configurations, different locations, okay? An isotype is the type of antibody found in all different individuals in a related species, while the allotype are the types of antibodies within a species, and the idiotype are the types of antibodies found within a specific individual, okay? So, again, not even in identical twins, all right, are you going to end up finding that all the immune cells are exactly the same in terms of genetic sequence, okay? Because rearrangement at the DNA level is basically a random process until a functioning gene is produced, and then the rearrangement stops, okay? We'll talk about the differences between primary and secondary antibody response, and talk about B cell differentiation and B cell receptor, and talk about how the immune system generates antibody diversity, and talk about the role of memory Bs during antibody response. So the first thing we want to address is something called clonal selection, okay?
Each B cell circulating throughout the body or harbored in a lymphoid organ is programmed to synthesize antibody that reacts with a single epitope. In a process called clonal selection, the antigen selects which B cell clone proliferates and differentiates into antibody-producing plasma cells or memory B cells. B cells display their antibodies on their cell surfaces. Antigen binding activates a naive B cell, a B cell that has not previously encountered antigen, leading to its proliferation, or clonal expansion. A single B cell can proliferate into many offspring. Some of these offspring differentiate into plasma cells. Plasma cells produce and secrete large amounts of antibodies specific to the original activating antigen. Others differentiate into memory B cells. Memory B cells do not secrete antibodies themselves, but are primed to quickly become antibody-secreting plasma cells if the host is reinfected with the same pathogen in the future. These photos show a resting B cell, before activation and proliferation, and a plasma cell that is producing antibodies. Notice the increased size of the plasma cell compared with the B cell. The plasma cell cytoplasm is stuffed with endoplasmic reticulum needed to support the incredible increase in protein synthesis associated with antibody production.
Okay, so the primary antibody response is just what happens during the very first encounter with a particular antigen, okay? A B cell gets activated by seeing the antigen that it's supposed to recognize, and then by interaction with a helper T cell, it becomes fully activated and turns into a plasma cell and begins secreting large amounts of antibody, and also dividing and producing, in some cases, memory cells, which will stay with the individual their entire life. The first class of antibody produced in this process is the IgM in the blood, and that will be followed in the secondary response by the IgG. Okay? In clonal expansion, only the B cell specific for that antigen is going to be stimulated to divide, and the rest are going to be left alone. Some cells are kept in the lymph nodes as memories, and we talked about IgM and then switching to IgG. Okay, so the way that happens is through a process called alternate splicing. Okay, we basically latch a different constant region onto the variable region by changing the way that the messenger RNA gets its exons hooked together. Okay, so you have more exons than you need, and then when you do the switch, you skip exons that are important in making the IgM constant chain, and you use exons that are critical in making the IgG constant chain, and you leave the variable region exons alone. Okay. So you can see here the process, right? The first thing that happens is a naive B cell, which is made every day in the red bone marrow, wanders around and then finds its antigen. Okay, so bingo. Oh, I see something I'm supposed to bind to. All right, the antigen binds the B cell with the antibody, and this cell may or may not require a T cell to begin expansion. If it requires T cell interaction, what will happen is it'll internalize this and it'll present it with an MHC, and then T cell and a B cell will buddy up, all right, and that second interaction will result in clonal expansion and then differentiation into a plasma cell, and then we whip out all kinds of antibody, and then we store memory cells during the process. Okay. In the secondary response, after we've activated the B cell, memory cells are made, okay, and when the memory cells see that antigen again, they immediately produce in large quantity, all right, the more effective antibody class, the IgG, and we do it over a much shorter time frame, okay? And that's why generally you don't get sick a second time with the exact same strain, okay? B cell receptors are membrane-bound antibody. Okay, remember that this would be the class that is capable of interacting directly with the B cell membrane. So it's a B cell receptor, and these, I believe, are the IgDs. Okay, antigen binding sites, once they encounter their antigen, cause these B cell receptors to cluster, to cap, okay, and then once they get internalized, that B cell begins to divide and make many copies of itself, and also process and present that antigen on its surface in the event that it needs to be activated by encountering a helper T. Okay, the capping is just the binding of the receptors on a B cell with a large repeating epitope, while the helper T is for T cell dependent activation of the B cell. So it's a two-step process, right? We encounter our antigen, so that picks out the B cells that are going to be effective at attacking, and then we encounter the helper T so that it stimulates only those B cells that have already been in contact with their antigen and no others, okay? And the result then is that turns it into a plasma cell, and the antibody production goes way up, okay? And we talked about the reason for all of this different, what we call antibody diversity, okay? And this has to do with what we call germ level DNA recombination, okay? Essentially what happens here is that when the immune system is being formed, pieces of DNA at different locations on the chromosome get recombined with each other at the DNA level, at the chromosome level, and it's only when we make a functioning heavy or light chain gene, okay, or a functional T cell receptor gene, that the rearranging stops, right? And now that particular clone is going to make only those antibodies or those T cell receptors. The next process selects among that population for those T cell receptors and antibodies that can recognize foreign antigen and destroy it, and then leave our own antigens alone. So we call that tolerance and specificity. Okay, and those that flunk the test are either destroyed or they're rendered in a state of anergy, where they're alive but not participating in the immune system. Okay, so these white blood cells, these lymphocytes, break the rule that says in your body, if we were to look at the DNA in your body from front to back, all 46 chromosomes, that the sequence would be the same going from each chromosome all the way through. Okay, chromosome number 1, 2, 3, 4, 5 are the same in every body cell with the exception of your lymphocytes, specifically in the regions where we code for T cell receptors and antibodies. Okay. We'll talk about the different types of T cells and their role in immunity. We'll talk about MHCs, talk about antigen processing, and describe what autoimmunity is all about. Okay, so in the thymus, right, what happens is that cells that are eventually going to become T cells migrate from the red bone marrow into the thymus in order to be processed for tolerance and specificity, right? And if you flunk that, you're either destroyed or rendered into anergy, okay? And then those cells that have passed the test are released out into the body, and they go out and find the tonsils, the lymph nodes, the malt, the gall, all that stuff that we talked about earlier in the lymphatic system, okay? In the T cell population, the cells that carry the receptor CD8, those are called cytotoxic T cells, and CD4 are helper Ts, okay? So what does a cytotoxic T cell do? It goes out and destroys infected cells and tissues by punching holes in it and spraying it with chemicals that cause apoptosis. What does a T helper cell do? Well, a lot, okay? It finishes the activation of B cells into plasmas, and it finishes the activation of cytotoxic Ts into fully active effector cells, ones that actually go out and do the work, okay? So if you lose your helper T population, you lose an effective immune system. And that's one of the reasons that AIDS is so devastating, because it attacks the helper T cell population. Okay? So T cell activation requires antigen presentation through a macrophage, okay, or some other antigen presenting cell. Okay? What will happen is that class 1 MHC protein complex with foreign antigen is basically a signal for that cell to be destroyed, okay? But it can also serve as a way to activate a wandering cytotoxic or a wandering B cell, okay? The first stage of activation. The class 2 MHC is what's found on a macrophage or dendritic cell, and when it presents foreign antigen, that is a way to stimulate the helper T cell in order to prepare it to activate already activated B and cytotoxic Ts, okay? Those that have encountered their antigen for the first time, okay? So not naive ones, but ones that have bumped into the antigen they're supposed to recognize, okay? So it's like a two-step process in most cases, right? The B is wandering around, it sees the antigen it's supposed to interact with, it binds it, okay, on its B cell receptor, and internalizes it, and then it presents it on its surface with an MHC, and then that is going to go and interact with a helper T, and that helper T, which has already been activated by interaction with perhaps an APC, right? Or even if not, if this interaction with this antigen from the B cell is its first encounter, what will happen is that hookup, okay, because the antigen is binding both receptors on the surfaces of both cells, is going to result in the complete activation of that B cell. So there's a physical hookup that occurs between the helper T and the B, right? With the cytotoxic, it's a little different, okay? In the cytotoxic T cells, they encounter their antigen for the first time, so they're ready to go, and then it's the helper T cell producing diffusible factors, like cytokines, that finishes the process of activating those initially activated cytotoxics, and then they go out and become effector cells. Okay? So let's look at antigen processing and presentation.
An antigen-presenting cell, or APC, is a phagocytic cell that degrades large antigens into smaller antigenic determinants and places those determinants on its cell surface for presentation to T cells of the immune system. Antigen presentation occurs on membrane proteins called major histocompatibility complex, or MHC proteins. The assembly of MHC proteins, of which there are two kinds, MHC1 and MHC2, begins in the endoplasmic reticulum, or ER. Antigenic determinants are attached to MHC proteins in two ways. MHC1 is used to present intracellularly synthesized antigens, and MHC2 is used to present antigens synthesized extracellularly. Endogenous antigens are those synthesized by viruses and intracellular bacteria as they grow within the cytoplasm. Some proteins that are produced during an infection are degraded by complexes called proteasomes inside the cell. The proteasome cuts the protein into small peptides, the antigenic determinants, which are then transported into the ER and loaded onto MHC1 proteins. In the ER, the peptide binding site on MHC2 is blocked by the association of a protein called the invariant chain, so only MHC1 carries antigenic determinants originating from intracellular antigens. MHC1, with its antigenic determinant, buds from the ER in a transport vesicle that fuses with the Golgi complex. From the Golgi complex, the MHC1 protein, with its peptide, is delivered by a secretory vesicle to the cell membrane, where the APC can present the intracellular antigen to T cells. MHC2 proteins, with their associated invariant chains, also bud from the ER and fuse with the Golgi complex. From the Golgi, MHC2 proteins then move into late endosomal compartments, which have proteases for digesting the invariant chain. The MHC2 protein is now poised to bind to antigens. Antigens produced outside of the APC, called exogenous antigens, will enter the cell via phagocytosis. Most bacterial antigens originate outside the cell and are internalized in this way. Also, large proteins, carbohydrates, and other molecules are engulfed, processed, and presented on surface MHC molecules. The endocytic compartment contains enzymes that begin to break down the exogenous antigen, producing peptide antigens. The fusion of the two compartments allows the peptide antigens to be attached to MHC2 molecules. The MHC2 protein, with its peptide, is carried to the cell surface. The stronger the binding of the antigens to these MHC surface proteins, the easier it is for T cells to recognize the complex. Once decorated with antigen, the professional antigen-presenting cell travels to lymph nodes where it presents its antigens to T cells.
Okay, so here we're looking at antigen processing and presentation. Class 2 presentation, the pathogens are phagocytized, broken down, and MHC2 foreign antigen complex is presented on the surface. These cells that are doing this are not going to be destroyed by the immune system, but instead are going to warn the immune system of the presence of an invader. Okay? Class 1 MHC, foreign antigen complex with class 1 MHC, is a signal for that cell to be destroyed, but it can also serve to activate a naive B or cytotoxic T. Okay. T cell education in the thymus, we talked about this, right? In positive selection, T cells that recognize self-MHC are allowed to live if that recognition is weak, okay? And in negative selection, T cells that bind to self-MHC too strongly are going to be wrecked, okay? So we don't want that hard hookup, right? Because that hard hookup is a signal to nuke the cell, okay? We want a weak hookup. That's the tolerance part, okay? The tolerance part. And then we go through the process of picking out cells that recognize foreign antigen, okay? And if they do, then they are are able to pass into the fully developed immune system. Regulatory Ts block the activation of self-reactive lymphocytes, okay? And this is one of the guards against autoimmunity, where the immune system turns on its own tissues. And there are several theories out there about how autoimmunity works. So what we're going to look at now is the hookups that actually lead to the activation, okay? So T cell receptor is found on T cells, okay, regardless of the type, right? Cytotoxics, helper Ts, regulatory Ts, and memory Ts all have T cell receptor, okay? They bind the antigen on the surface of the T cell. So the T cell receptor binds peptides within the MHC, and it is also associated with the antigen, okay, and also associated with something called CD3, okay. So when it's going to hook up to whatever it's going to interact with to finish the activation process, okay, the antigen will be like the middle of a sandwich between the T cell receptor and the MHC, okay. So it goes T cell receptor, antigen, MHC, right? And then you've got cell 1 on one side and the T cell on the other side, okay? And then CD3 is a way to ensure that hookup is a solid one, okay? So CD3 kind of reaches across and interacts with MHC, okay? So a helper T that's naive is going to bind an antigen-presenting cell that has foreign antigen on MHC2. The CD4 receptor on the naive T helper binds to the self-MHC2, okay? And the CD28 on the T cell surface also binds to B7 on the antigen presenter, and that activates the T helper, okay? And now that activated T helper is ready to activate B cells that are not naive and cytotoxic Ts that are not naive, okay? So there is the interaction that takes place, right? The bone marrow kicks out the precursors which migrate to the thymus, which then get selected in positive and negative selection to make up the helper T complement of the entire immune system. And then we take up residence in the secondary immune organs like the lymph nodes, the tonsils, the spleen, okay? And then what we do there is we encounter antigen-presenting cells, okay? Antigen-presenting cells like macrophages hook up to the naive T helper. You can see the hookup here. See how CD28 and B7 meet, and how CD4 is hooking up with MHC, right? So this is T cell receptor here in red and orange, and this is MHC over here in dark green and light green, okay? And then what happens is that helper T gets clicked on, and now it can turn around and using T cell receptor and interacting with MHC2 on the B cell, it can activate the non-naive B, okay, and then it can become a plasma cell and go out and do its work, okay. The plasma cell derived from the B cell is the antibody producing factory, okay, while the naive B cell has to encounter and cap and take in the antigen using B cell receptor and then present it on its surface with an MHC2, okay. So there's physical contact here in order for this to happen, right? Whereas cytotoxics don't require physical contact with the helper T, okay. And then memory Ts, of course, are produced throughout the process, okay. When an activated helper T meets the B, the antigen cross-links the receptors on the membrane, right? So you've got that sandwich of MHC1, antigen, and then, not MHC1, sorry, MHC2, antigen, and then T cell receptor, okay? And then those other proteins that help to hook up the firm, right? CD4 and so forth. During the early phase of the primary response, the second signal is the binding of complement factor C3 bound to pathogen, and during the late phase of primary response, the second signal is the binding to the T helper, okay? And this causes CD40 to bind CD40 ligand on the surface of the T helper. And once activated, the helper T, by the helper T cell, the B cell can do all the production of all those different flavors, all those different classes of antibody, all of which bind the same antigen, right? So it can switch from IgM to IgG to IgA and so forth, right? And remember that these are all found in different locations, right? IgG and IgM in the blood, IgA in things like milk and colostrum. We'll talk about how cytotoxic T cells get activated, talk about how activated cytotoxics recognize their host, talk about cytokines, talk about antigens and superantigens, and talk about ways that pathogens get around immune function, okay? So with cytotoxic Ts, right, the cytotoxic T binds to MHC1 antigen complex, okay? The T cell receptor is specific for that antigen, so that's our first start in the activating cascade. CD8 on the cytotoxic T binds self-MHC1 on the target, okay? And IL-2 produced by helper T binds to the cytotoxic T, okay? But that's only going to happen if that helper T has already been activated, okay, by encountering its antigen, okay? And then that second round of activation results in an effector cell going out and doing the work. So let's look at antigen processing and presentation.
Cell-mediated immunity is a type of adaptive immunity in which cytotoxic T cells, or TC cells, recognize and kill infected host cells. As with other lymphocyte types, cytotoxic T cells park themselves in lymph nodes. There they await interaction with an antigen-presenting cell, or APC, newly arrived from an infected site. For a cytotoxic T cell to become activated, its T cell receptor must bind to antigen presented on the APC's MHC1. At the same time, a protein called CD8 on TC cells attaches directly to MHC1. This binding between the two cells provides the first signal for TC activation. The second signal, which completes the activation process, comes from a cytokine called IL-2, produced by the Th1 class of helper T cells, which have also been activated during the infection. Upon binding IL-2, the TC cell gains cytotoxic activity, leaves the lymph node, and travels to the site of infection. It attaches to any cell bearing the same peptide MHC1 complex, for example, cells infected with the same virus that triggered TC production in the first place. All cells of the body, with the exception of red blood cells, express MHC1. Many MHC1 proteins on an antigen-presenting cell display bits of the cell's own proteins, but some MHC1 molecules display bits of the viral proteins made as the virus replicates itself inside the cell. By displaying viral peptides, this cell identifies itself as being infected. Cytotoxic T cells contain granules full of proteins called granzyme and perforin. After binding to the infected cell, the TC cell releases the contents of its granules onto the target cell. Perforin produces a pore in the target cell membrane through which granzymes enter. Inside the infected cell, granzymes trigger a cascade of events leading to cell death. Cell-mediated immunity works by sacrificing the body's own cells because it also kills the pathogens inside them.
So superantigens bind outside of the MHC binding site and generate cytokine production, okay? They're not antigen-specific responses, and a lot of different T cells can be activated as a result, okay? Normal antigens are going to bind on the left side of this figure, right? And the superantigen is binding as shown on the right side, okay? So see the difference, right? Normal antigen inside the sandwich, super antigen outside the sandwich, right? So this puts this whole interaction into overdrive, okay? So you can see the cytokine, the sample source, and the general functions. Okay, I won't go through every one, but you can review that. It's table 16.4 in your book. So how can pathogens escape immune function, right? Some viruses can downregulate MHC1 or can use a decoy MHC to distract the immune system. Some pathogens change their surface proteins through variation, so they keep putting on a different disguise. Some pathogens cause the death of T cells. An example would be AIDS, right, which attacks the helper T cell and turns it into a virus factory so that it can't do its job. And then some block cytokine production that produces inflammation. And all of that can cripple the immune system, okay? Now, these are some of the videos you've looked at. What I want to show you next is just a sort of a summary of immune function, all right? And then I will join everybody in the next podcast. I thank you for listening.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.